Outside my door. Okay. So welcome everyone. The purpose of these journal clubs is to reinforce some concepts that we've brought up in the workshop. So our last workshop, we covered metabolic resistance to insecticides and copy number variation. And today, Eric Lucas will present his paper on whole genome sequencing reveals high complexity of copy number variation at insecticide resistance loci in malaria mosquitoes. So Eric, I'll let you introduce yourself and go ahead.
Great, thank you very much. So hello, so yeah, I'm Eric Lucas. You possibly remember me from the second workshop where I gave one of the lectures on the methods for detecting CNVs in the AG1000G data. So I'm a postdoc at the Liverpool School of Medicine, and I wrote the pipeline to discover CNVs in the AG1000G data, and that resulted in an initial paper in 2019 where we reported the findings of applying that to phase 2 of AG1000G. And since then, I've sort of been maintaining that pipeline and sort of having oversight of the CNV data from AG1000G. And so Alistair asked me to give a sort of presentation of that 2019 paper to run through, because so far you've sort of seen what the methods are for detecting CNVs and how you can access those data from the MalariaGEN API. But I think we thought it would be interesting for you to see as well the sort of results that we get and the sort of interpretations that we've made from those results, as this could be applicable to further research on the topic. So I'm going to sort of assume that you were in the workshop, and so I'm not going to run through all of the same background that we did before, but just a background. So we know that metabolic resistance is an important mechanism for insecticide resistance, and we know that overexpression of certain genes is associated with increased metabolic resistance. So notably from the literature on gene expression studies, we know that, for example, CYP6P3, GST2, CYP9K1, and CYP6M2, amongst others, have been sort of consistently coming up in gene expression studies comparing resistant and control mosquitoes. And what we don't know is what genetic mutations cause this overexpression. And there are a lot of different types of mutations that can cause overexpression, not just CNVs. So changes in expression could be caused by changes in the nucleotide sequence in the regulatory region. It can be caused by other changes in the surrounding DNA. It can be caused by mutations in completely different parts of the genome that code for what's called transcription factors, which are in turn affect the expression of the gene that we're interested in. But CNVs is one of the mechanisms by which increased expression could occur. And so we wanted to sort of look for CNVs across the Anopheles gambiae 1000 genomes dataset to get an idea of their variety and their distribution. So just a sort of quick recap of the history of AG1000G. So we're talking about whole genome sequence data from thousands of individuals of Anopheles gambiae, but also Anopheles coluzi from sub-Saharan Africa, to study the population genetics and also insecticide resistance. Phase 1 consisted of 765 individuals, and that was published in 2017. Phase 2 consisted of 1,142 individuals, and the results from that were published in 2020. Phase 3 has 2,783 individuals, and obviously we're sequencing more and more individuals, including a lot of PAMCA samples subsequently. But the results that I'm going to talk to you about today come from phase 2. So that's 1,142 individuals from across West, Central, and East Africa. And these are all Anopheles coluzi and Anopheles gambiae. There were no Anopheles arabiensis in this data set. And so the question we wanted to ask was, what is the importance of gene duplications in the evolution of metabolic insecticide resistance in Anopheles gambiae? So we wanted to discover CNVs across the genome and across all of our study populations and get an idea of CNV diversity. So you may remember from the workshop that one way in which we discover, in which we can detect CNVs is by using the reads that come from this CNV breakpoint. But this, as you may recall, is quite laborious. It requires quite a lot of manual curation. And so we can't do this across the genome. It has the advantage that we can tell apart individual CNV alleles from each other, but we can't do it across the genome. And we wanted a method that worked across the genome. So we used the coverage method, which again, hopefully you'll remember from the workshop, where essentially we calculated sequencing coverage across the genome, and we applied a hidden Markov model through those data, and we identified regions of concerted increased coverage. And we did this on an individual level. So for every individual, we ended up with an idea of what CNVs they carried. However, this method is to some extent prone to error, and we wanted to be, if we're interested in the sort of the distribution and the evolutionary history of CNVs, then we want to have some idea of shared CNVs, so of CNV alleles, which we could get from the breakpoint data, but from a genome-wide perspective, we needed to get some idea of this from the coverage data as well. So how can we match how can we get increase the quality of CNVs that we find to sort of remove ones that are potentially due to error? And how can we match CNVs across individuals? So one way in which we increase the quality of CNVs was to require a minimum number of, a minimum size, genomic size of the CNV. So we call the CNV if the estimated copy number state from the hidden Markov model was above baseline for at least 5 consecutive windows, and each window was 300 base pairs. So here in this hypothetical situation, we would call this a CNV in this particular sample, but here we would not, because it only spans 4 genomic windows. So by removing very small CNVs, we can hopefully remove artifacts that are just due to very sort of transient increases in coverage. We then matched CNVs between individuals based on where the changes in coverage occurred. Now, the problem is that because of the random noise in the data, the same CNV in different individuals might not start and end at exactly the same windows. There might be a little bit of variation between individuals as to where the hidden Markov model decides that the coverage increases or decreases. And so what we did was we said that a CNV in 2 different individuals was the same if it was within 1 window of each other. So this increase in coverage here happens 1 window away from this one, and it ends at the same place. This one ends 1 window away and begins 1 window away from this one. So we class all 3 of these as the same CNV. And then to determine the official start and end point of the CNV, we just take the average of all of the start and end points. In this case, we took the average in the form of the median. And so in this way, we could say across individuals which ones have the same CNV, and we can get a hopefully more accurate idea of exactly where the CNV starts and ends. And we can then line that up with the positions of known genes in the genome to say, okay, well, this CNV covers gene Y, for example. And so all 3 of these individuals have a CNV in gene Y, but it only partially covers gene Z, for example. And what this meant that we could also filter CNVs by frequency. So we could say, okay, in order to make sure that we don't have that we reduce the number of incorrect CNVs, we want to find them in more than 1 individual. So if we find a CNV in just 1 or even 2 individuals, we say, well, that's not enough evidence that this is a true CNV. It might be true, but we're not sufficiently confident that it's true. So we required CNVs to be found in at least 5% of individuals in any given population in the data set. And so again, this wouldn't be possible if we hadn't matched these CNVs between individuals. So the results then, we found a total of 1,557 CNVs that passed all of our quality filters. And together, these 1,557 CNVs covered 267 genes. And this is already a significant enrichment. So CNVs were more likely to be in regions containing genes than otherwise. If these 1,557 CNVs had been randomly distributed across the genome, we would expect to find fewer than 267 genes. So already CNVs are enriched for genes. 27 CNVs were in regions of metabolic genes, and we called metabolic genes anything that was a P450, a GSTE gene, or a carboxyl esterase. And again, this was a significant enrichment. So even if we take into account the fact that the CNVs were more likely to cover genes, if we randomly assign CNVs to genes, we would end up with fewer than 27 metabolic genes. So finding 27 CNVs in metabolic genes was a significant enrichment. So we have enrichment of genes, and within that we have enrichment of metabolic genes. And 16 of those 27 CNVs cover one of the following 6 genes: CYP6AA1, P3, M2, Z1, GST2, and CYP9K1. And if you're familiar with the metabolic gene literature, you will recognize this as a who's who of genes known to be involved in metabolic resistance. And that's, over half of the 27 metabolic genes that we found are ones that we have really strong evidence from the literature that they are involved in metabolic resistance. So at every level, we're seeing enrichment of CNVs around genes that have reason to be involved in metabolic resistance. We can also take another approach. We can ask whether genes covered by CNVs were enriched for certain gene ontology terms. So in every gene that's annotated in the Anopheles Gambia database is assigned a list of gene ontology terms, which is just a series of tags that say, this is a process that is associated with this gene. This is something that the gene is involved with. And we can ask whether any of these tags are more likely to be found in genes covered by a CNV than random. And we found a list of, I think it was 13 gene ontology terms that were enriched in CNVs. And at least 4 of those gene ontology terms are associated with P450 activity. So these 4 terms are all ones that are associated with cytochrome P450s. And another group of these Go terms were associated with peptidase activity, so proteins that degrade other proteins. And this, we're not sure whether it's associated with insecticide resistance, but potentially it could be. So the next thing that we found was that CNVs are at really high frequency in some populations. So here are those 6 genes that I showed you earlier on the rows in this table. And here are all of the different populations that we found that are present in the phase 2 data set. And here are the proportion of individuals that have a CNV in each of these genes in each of these populations. And darker red indicates higher proportions. And you can see we have really high frequencies in some places. So over 90% of mosquitoes carrying CNVs in some of these genes, in some of these populations, and quite a few where we have over 50% frequency. So we're seeing really high frequencies of CNVs. And the next question we wanted to ask was, well, how many independent origins of these CNVs are we seeing? So when we see, for example, CYP6AA1 with such really high frequency in different populations, is this a single CNV that we're finding in all of these populations, or do each of these populations carry different CNVs? And within these CNVs, within these populations, are we seeing more than one CNV present in the population at any given time? And so it was this that made us decide to use these CNV breakpoint reads in order to get a much more precise idea of exactly how many different CNVs we found in the data set. And what we found was that there were a lot of CNVs, and again, they were concentrated on the genes involved in metabolic resistance. So this is the cluster of GSTE genes, and we found 11 separate CNVs, which we call DUP1 all the way through to DUP11. And this each row here is one of the different CNVs that we found, showing you where it starts and where it ends. So the region covered by this bar is the region that's duplicated by the CNV we've called DUP1. And the gene in this cluster that is really strongly associated with insecticide resistance in the literature is GST2, which I've highlighted in red. And you can see that 10 out of the 11 CNVs all cover GST2, which is not something you can say about any of the other genes in the cluster. So we again have a real concentration of CNVs in this gene most strongly associated with resistance. And we found the same thing in other clusters. So this is the CYP6P, the CYP6P, CYP6AA cluster. And And this is the gene that's most strongly associated with resistance in the literature. And we found 5 different CNVs covering this gene. But interestingly here, the majority of CNVs are in the gene CYP6AA1, which before this paper was most strongly associated with resistance in other species such as Anopheles funestus. And here we saw 13 out of 15 of these CNVs covering CYP6AA1, suggesting that actually maybe this gene had a more important role in insecticide resistance in Anopheles gambiae than we previously suspected. And these maps show for CYP6P3 in red and for CYP6AA1 the countries in which we found the presence of these CNVs in the data set at appreciable frequency. So CYP6P3 was very much concentrated in West Africa, whereas CYP6AA1 was more widespread in West Africa and also present in East Africa. So next we wanted to ask, are these CNVs under selection? So is there any evidence that these CNVs are spreading rapidly in the population, which would be an indication of positive selection? So I wasn't in the last journal club, I'm afraid. So Chris Clarkson may have already covered some of what I'm about to say in his presentation, but I don't think that a little bit of repetition is a bad thing. So how do we detect the presence of selection? Well, so here we have, imagine a mosquito, and this is one of the chromosomes that mosquito carries. And when that mosquito has offspring, so it will have one copy of this chromosome from its mother, one from its father. Let's assume that this orange one is the one it got from its mother. And when it has offspring, it will pass on that chromosome, and that chromosome will recombine with the other one. So the one it got from its father is shown here in blue. And so this is one offspring that it's had, this is another offspring that it's had, and those offspring receive a mixture of each of this mosquito's chromosomes at this location. But you can see that there are big chunks of one chromosome followed by big chunks of another. So, it's given this first offspring, almost 2/3 of this chromosome is the orange version. And again, here, almost 2/3 is the orange version. So this transmission, this breaking up of the chromosome happens in big chunks. But then each of these offspring will go off and have offspring of their own. And this is going to get broken up again. So here it's being broken up with this purple chromosome, here it's being broken up with this yellow chromosome, and so on through the generations. At each generation, it gets broken up more and more. And so the more closely related two individuals are, here we have some full siblings, whereas here we have some very distantly related cousins, the bigger the chunks of DNA that they share in a row. And so if you look at a given mutation of interest, for example, let's say that we have a CNV here that we're interested in, then if you compare that region between two very closely related versions of this chromosome, there are very big chunks of shared DNA either side of it. Whereas if you compare it in two versions of this chromosome that are much more distantly related, then there's much smaller chunks of shared DNA either side of it. Okay, and this is essentially what we look for when we try and look for positive selection. Here I'm comparing 6 generations to 1 generation. In reality, what we're doing is comparing hundreds or thousands of generations to maybe millions or hundreds of thousands of generations. But essentially, the process is that it's looking at, it's using the fact that more closely related versions of the chromosome will have much larger chunks of shared DNA around a single shared mutation. Okay, and so this is what we found. So here we're looking at some of the various CNVs in CYP6AA1, so DUP1, DUP7, DUP10, etc. And we're looking at the length of that shared chromosome to the left and to the right of that CNV. And we're comparing it against some wild type, i.e. some non-CNV chromosome. So the same area of the chromosome, but in samples that don't have the CNV. And here you can see much smaller, much shorter lengths of shared haplotype when we look at the non-CNV regions compared to when we look at the CNV regions. And so this is telling us that these CNVs are under strong positive selection. Essentially what it's telling us is that 2 copies of this, of the CNV drawn from the population are quite closely related. They came from a common ancestor relatively recently. whereas 2 copies of a non-CNV DNA at the same region came from a common ancestor much, much longer ago, and so haven't spread as recently through the population. So what we have is that relatively recently, one of these CNVs occurred somewhere and then have rapidly spread through the population, so that any 2 CNVs chosen at random are actually fairly recently related. And this is just another way of looking at the same data. Here, again, we take a given CNV that we're interested in, or a given region of the chromosome that has CNVs, and we ask, as we move away from that CNV on the chromosome, how quickly do pairs of individuals stop looking like each other? And if those pairs of individuals are do not have the CNV, they very, very quickly become dissimilar as we move away from that region of CNV. Whereas if individuals have a CNV, then it takes longer before they become less similar as you move away from the genomic region with that CNV. And this is called extended haplotype homozygosity. So in summary of this paper then, CNVs in the Anopheles gambiae genome are enriched for genes, and in particular for genes involved in metabolic insecticide resistance. The CNVs in key metabolic genes are found at very high local frequencies. Often over 90% of mosquitoes have a CNV in these genes involved in metabolic insecticide resistance. In the metabolic gene clusters, we see many independent origins of CNVs, and particularly these tend to be focused in the key genes that we know to be important from the literature. And the CNVs are spreading rapidly under positive selection. So thank you very much. And if you have any questions, or if you have any thoughts on the paper, or any sort of musings, then please fire away. And thank you for listening to the quick presentation.
Thanks a lot, Eric. Any questions? Very welcome in the chat box, or you can just raise your hand or speak up. I could start with a question. Oh, we have one. Please comment on minimum sample sizes of mosquitoes required for this type of analysis.
So it depends. It depends which sort of question you're asking. So if we want, so for the selection analysis, it's hard to tell, and it will depend on how strong a signal that so how strong the selection is. The stronger the selection, the small, and how frequent the CNV, then the smaller the sample size that you need. We had sample sizes of sort of 40, 50 individuals per population, and that proved to be sufficient. You would probably struggle with sample sizes of less than 10, I would say, but it really does depend on how strong is the signal that is there to be detected. And then in terms of finding the diversity of CNVs, then again, obviously, if you only have 2 individuals, then that's 4 chromosomes, because each individual has 2 chromosomes at each locus. So you're not going to find more than 4 CNVs. So the more individuals you have, and particularly the more different populations you sample from, the more of an idea of the diversity of CNVs that you'll find. So in particular, for example, in this cluster, the CYP6AA and CYP6P, we found 15 CNVs out of these 1,152 samples from these countries. But then in phase 3, with roughly double the number of samples from some more different populations, that doubled. We now have almost 30 CNVs that we are aware of. So the more different places you look at and the more individuals you sample, the more CNVs you're going to find. However, a lot of the extra CNVs that we found are very low frequency. And what that tells you is if you have a CNV at very high frequency, you're not going to need to sample, at 90% frequency, for example. Probably if you sample 2 or 3 individuals, you're going to find that CNV. If you have a CNV at sort of 1% to 5% frequency, you need to have a much larger sample size before you detect that CNVs. And so as we increased our sampling, we found all of these new CNVs, but a lot of them are at quite low frequency, because the high frequency ones, apart from the ones that were only in the new areas that we looked at, the high frequency ones we had already found. So it really depends on sort of what you want to do Similarly, you know, the impression that the idea of how common these CNVs are, you don't need a huge sample size for that, because the very high frequency ones, you know, this 90% frequency CNV, if we'd had a sample size of 10 here instead of 75 mosquitoes from Burkina Faso, we'd probably still have found that CNV, and we'd have a reasonably good idea that it was a high frequency, around 90%. Yeah, I think does that answer the question? Unfortunately, the answer is that it depends, but that hopefully that's given you a sort of ballpark figure of the sample sizes that you would need.
So we have questions rolling in. Albert asks, how do you compute the p-values of CNVs for the metabolic genes?
You might have to elaborate. So you'd have to say exactly what question you're, so do you mean, so okay, sorry, do you mean this one? Is this the p-value that you're referring to for the enrichment of metabolic genes?
I think so. They said 27.
Okay, yeah. So this was done, we did this in a slightly more complicated way in order to control for the fact that CNVs cover, you know, a single CNV can cover several genes. So for example, you know, this DUP10 covers 4 different genes. And so the fact that all 4 of these genes are have a CNV in them, they're not independent sample points. So we can't say, hey, look, there's this CNV in CYP6AA1 and also one in CYP6AA2, because it's just caused by the fact that there's a single CNV and that these genes occur in clusters. So we had to account for this clustering of genes on the chromosome. So the way we did it was we took each CNV and the number of genes that it involved. So for example, this CNV covered 4 different genes, and then we randomized those 4 genes in the genome. So we just said, okay, let's take another group of 4 genes all next to each other, completely randomly determined in the genome. And we do that for every CNV. So for every CNV we found, obviously from the coverage data only, because we did it genome-wide, for every CNV that we found, we identified how many genes it contained, and we randomized where those genes were in the genome, and that gave us an idea of the expected number of metabolic genes that we should find. We did that 10,000 times, so we repeated that randomization 10,000 times, and that gives us an idea of the distribution of that expectation. And then we asked how many out of those 10,000 randomizations, what proportion of them had as many as 27 CNVs in metabolic genes, or had as many as 27 CNVs in metabolic genes. And the answer was none of them. So none of those 10,000 simulations, randomizations, had as many as 27 CNVs that covered at least one metabolic gene. And so this p-value is smaller than 0.002. That's drawn from a two-tailed test, which is why it's 2 instead of 1. And so if there had been 1 out of those 10,000 randomizations that had at least 27 CNVs, then the p-value would have been equal to 0.0002, because precisely 1 out of 10,000, our estimate of the probability of getting this value would have been 1 in 10,000, and because it's two-tailed, we double that, and it's 2 in 10,000. Because we had none, then all we can say is that p is smaller than 0.002. We can't get a more accurate p-value without doing even more randomizations. But p smaller than 0.002 was a sufficient level of precision for us. So hopefully that's answered your question. Please let me know if it's unclear.
Al-Haji asks, regarding selection around CNVs, do we know anything about the source of selection pressure? So does anything in the metadata indicate increased vector control interventions or agricultural activities with insecticide use in these different regions that you're comparing?
So not from the 1,000 genomes data. However, there are some follow-up studies that we've done that show that at least some of these CNVs, the CNVs that we've focused on, are correlated with insecticide resistance. So if you take individuals that have or don't have one of those CNVs, the ones that have the CNV are more resistant. In this case, it was CYP6AA1, and individuals that have a CNV in CYP6AA1 were more resistant to deltamethrin. Now, whether or not that's due to insecticide use or agricultural use, no, we don't know for sure what the driving factor was in the environment to lead to the spread of those genes, of those CNVs, sorry.
Mugenzi asks, is it possible to determine the impact of this CNV, maybe that's just any general CNV, with insecticide resistance? So how do we link one CNV with insecticide resistance?
Yeah, that's a really good question, and it's very tricky. You know, we as I say, there are so many of these CNVs, and we're left sort of analyzing them one by one. So we spend a lot of time analyzing, for example, this CNV DUP1, which is found in Uganda. And that was a very painstaking study, and it was eventually found to be associated with deltamethrin resistance. And then we're left assuming that these other CNVs are also important, but we don't necessarily know that that's the case, and maybe they're not. Certainly with DUP1, we know there were other mutations nearby, and we don't know to what extent is DUP1 responsible for the increased resistance, or the other mutations next to it. And if it's more of the mutations next to it, or if exactly where the CNV starts and ends is important, then these other CNVs might be less important than DUP1. So it ends up being, we have to make a sort of decision as to at what point do we say, well, we're not going to believe a CNV is important until someone has studied that exact CNV. Or at what point do we say, well, we found that several of these CNVs are important, let's just assume that all of them are, even if we don't know exactly. But at the moment, we're not at that stage. We haven't analyzed enough of them closely enough to be able to sort of make that kind of judgment.
Elijah asks, do CNVs co-occur with SNPs? And do you see any form of positive selection of both forms of mutation, so target site resistance and CNVs, and their role in metabolic resistance?
Yeah, no, absolutely, they do, and it's very tricky. So this sort of comes back to my answer to the previous question: is that it's very tricky to disassociate them. Quite often you find CNVs in your populations. You find, for example, that they're associated with resistance, but those CNVs do not exist on their own. They exist with a background of other mutations next to them, such as SNPs, or such as transposable element insertions, or such as anything else. And being able to disentangle those different mutations and what their effects are is very challenging. And in general, you can't do it by just collecting samples from the field if those two mutations are closely linked. So what we are hoping to do soon, what we will be doing soon, is to try and use CRISPR to try and put these CNVs on their own without their associated mutations into various colonies of mosquitoes, to try and see what the effect of those CNVs on their own are. But that's, we haven't done that yet, and that's going to be a lot of work, and it's down the line.
Mugenzi asks, what could explain the CNVs observed for the serine peptidase genes? And how do you explain the CNV distribution in West and East Africa, but not in Central Africa? But that might be a sampling bias.
Yeah, so no, those are two good questions. So the serine peptidase, honestly, I don't know whether or not it's associated with insecticide resistance at all. We haven't really looked into them. We sort of observed the result, and it wasn't immediately obvious to us sort of where to push the research. We haven't really got into exactly which of those serine peptidases have CNVs and what they do. So that would be an interesting avenue for research, but it's not one that we focused on. But the data are there if anyone wants to have a look. The second question, Central Africa. No, that's also a very good question. We don't know, but there are various sort of intriguing patterns in the data that might mean something, but we're not sure what they are. So, for example, in West Africa, in Anopheles coluzi, we see a lot of CNVs in the CYP6AA, CYP6P cluster. In fact, if I show the frequency table, there we are. So in Anopheles coluzi in West Africa, we see a lot of CNVs in this CYP6AA, CYP6P cluster. We don't really see them in Gambi. We see much lower frequencies in Gambi in West Africa. What we do see in West Africa is very high proportions of CNVs in CYP9K1 in Anopheles gambi. So in Burkina Faso, the vast majority of coluzi have CNVs in the CYP6AA, CYP6P cluster, whereas the vast majority of gambi have CNVs in CYP9K1. And we don't know why this is, because when we switch to East Africa, then we start seeing, much higher frequencies of CNVs in CYP6AA1, but not a very high diversity, I should say. This is just one CNV that's reached very high frequency, whereas over here we're talking about a lot of different CNVs that are all reaching intermediate frequencies, and also, so back in Uganda we also have CNVs in CYP9K1. So why we're seeing these differences between East Africa and West Africa within Gambi, and why we're seeing this difference between coluzi and Gambi within the same locality, we just don't know. We're not sure what's going on. Why we're seeing less in Central Africa, again, we don't know. We're observing this. What the underlying processes are, we can't say for sure. For example, in Cameroon, we know that we've seen very strong signals of selection in the same loci. These same loci where we're seeing very high frequencies of CNVs in other populations. We see very strong signals of selection in Cameroon, but none of them are associated with CNVs. And so whether it's just that some other mutation has come in by chance and is already doing everything that needs to be done in that location, and so CNVs aren't getting the opportunity to spread because this other mutation sort of got in there first, we don't really know. But it's a really intriguing question, and it's one that we would love to have an answer to.
So while you're on this slide, the next question is, how do you compute CNV frequencies, and what is the threshold that you use to filter CNVs?
So I'm not sure what the second question is asking precisely. How to compute CNV frequencies? This is purely the number of individuals. So remember, all of the analyses I showed you were on an individual basis, right? So this, where's it gone? These CNV calls were made on an individual basis. This individual has that CNV. This individual has that CNV. This individual had that CNV. So for every individual, we can say, do you or do you not have a CNV in gene Y? And so then we can just say, well, what proportion of it within Burkina Faso and Anopheles coluzi, what proportion of individuals have a CNV in gene Y? And if gene Y is CYP6AA1, then the answer was 90.7%. So that's how we compute the CNV frequencies. The threshold, it depends what you mean. So in terms of the threshold to decide that a CNV is real, then it's if it was found in at least 5% of individuals in any of the populations in the data set. Now that's probably quite stringent. That's probably kicking out a lot of CNVs that are real, that will have been ignored because of this threshold, but we wanted to be really sure that we weren't believing any CNVs that were not real. So if a CNV is sort of an error, then hopefully that error wouldn't happen in more than 2 or 3 individuals. I should say one clarification, some data sets only have sort of 4 individuals in them, and so 5% is quite a small proportion of that. And so for small populations, we said we needed at least 3 individuals. So it needs to be found in at least 3 individuals within a given data set, unless the data set is much larger, and then it becomes 5% of individuals. So is that what you meant, whoever it was asked the question, is that what you meant by what was our threshold.
I think by the wording, they may have noticed that 5% at the end and then less than 5% in this table.
Oh, yes, sorry, so this is 5% if the CNV is present at 5% in a given population, then we believe the CNV in general. And then we add, so for example, here, there's a CNV at 0.7% in Cameroon, but that CNV is probably one that's also present maybe at higher frequency in, for example, Cote d'Ivoire. And so it stays in the data set because it's found at higher frequency in another population, but then we still believe the CNV. And so when we find it in Cameroon, we still consider that those individuals do have a CNV there. And so you end up with lower frequencies.
So we have a question from Catherine about which sequencing platform was used. And then the next question from El Hadji is, what about the fitness cost of CNVs? What do we know from the literature about the fitness cost of these CNVs?
So the first question, it's Illumina, and these were back for phase 2. It was 100 base pair paired-end sequencing, if I remember correctly. The more recent data is 150 base pairs. This one was 100 base pairs. What do we know about the cost? Basically nothing. But yeah, it's another interesting question to follow up on. So we do know, for example, in the CNV in ACE1, which isn't any of the ones that we looked at in this study, we know that there's a big deletion in ACE1 that and the CNV with a big deletion. So within the very large CNV in ACE1, there is a very large, some of the versions of that CNV have a very large deletion within the CNV, so that in the middle of the CNV, you revert to the normal copy number. And that CNV is I think almost more common than the non-deleted version, which suggests that CNV that deletion is beneficial, which means that the presence of the CNV in that deleted region itself carries a cost. But apart from that, yeah, no, we know very little about the costs associated with the CNVs.
Plenty of work to do.
Yes.
Albert asks, what do you mean by extended haplotype homozygosity? So could you expand on talking about extended haplotype homozygosity?
Yeah, so if you imagine again, so in this population here, we can compare these two individuals and we say, you know, how similar are they? And because we've only got one pair of individuals, then, you know, we can say, well, how similar are they in this direction? And the answer is completely similar, so 1. And then at this point, they become completely dissimilar, so 0. So if we just plotted this pair of individuals on that plot, we would get, you know, a value of 1 up to a certain point, and then it would drop down to 0. But then we average over many individuals. So we take many pairs, not individuals really, it's chromosomes, but many pairs of chromosomes found in the population. And we ask, you know, what proportion of them are the same? And so then you get this decline. So right at the point where where this pin is, because we're only looking at, you know, we take groups of individuals that are identical at the point of this pin. So if we take the color coding here, we're only going to look at individuals that are orange at the very point of this pin. So all of them are the same here. So all of them are 1. And, you know, this line, for example, is the ones that are orange, and this line is the ones that are some other color. So they're, you know, comparing them together. So they're always 1 at the start. And then you start getting smaller and smaller proportions. But because now we're, instead of making a single comparison, which would just be 1 or 0, we're taking, you know, what proportion of comparisons are still identical. We get this gradual decline. So if you just imagine a huge variety of these chromosomes, all of which are orange at this particular point, but all of which have some other arrangement of colors everywhere else, then we can just ask, you know, for the orange ones, if I'm here, what proportion of those individuals are still orange? What proportion is still orange here? What proportion is still orange here? What proportion here, etc. And so that's going to decline. And so you get this decrease. And so the haplotype, we use the word extended because we don't just look at a single point, we extend on either side. Haplotype, because that's what we mean when we look at a single chromosome instead of, you know, an individual has 2 chromosomes. So an individual if you look at an individual level, there'll be two alleles here. Maybe one of them's orange, maybe one of them isn't, and it becomes complicated to do this analysis. So we split it into haplotypes or chromosomes, and we compare individual haplotypes instead of comparing whole individuals with two chromosomes each. So extended haplotype, and then homozygosity in that they are all homozygous. What proportion of those comparisons are homozygous? Does that answer the question?
I think so. So there's a few questions about code availability. So about computing p-values, is the code available for doing these analysis?
Yeah, so the code is available from with the publication specifically for, I will have to double check. I'm pretty sure we made the code available for computing the p-values as well. But if I'm wrong about that, then you can certainly contact me and I will happily share any and all of the code that was used for this publication.
And Alhaji asks, is it possible to draw haplotype networks from your data? Were you able to do so? And what are the conclusions in terms of the origin of different CNVs?
So yes, it's possible. No, I didn't. Why I didn't, I can't remember. I think it just wasn't one of the things that we decided to do. We didn't have a particular, we didn't consider doing it and then decide, oh no, we can't because it just wasn't one of the things that we decided to do. So no, no, we don't have those data. What we did do, which is sort of similar, was identify the selective sweeps. So instead of just drawing those haplotype networks, which you probably saw from Chris's talk, we just, for each region, we said, how many big groups of haplotypes can we find and which individuals carry those? And so that can still tell it, that will tell us, for example, for a given CNV, does it belong to a single haplotype or does it belong to separate haplotypes? And what we found was that it's difficult because the haplotypes are not always perfectly reconstructed. And so if you find a mismatch, you don't know whether that's an indication that you're definitely seeing two different haplotypes or whether that's just an incorrectly reconstructed haplotype. But what we did find was that in most cases, there was a correspondence between a CNV and a haplotype. So a given CNV came from a certain haplotype. So it looked like, at least in most cases, we can't make a blanket statement about all CNVs, but in most cases, these CNVs are independent origins, where the ones that have different breakpoints have different origins. It's not just a CNV that then loses, has a deletion and loses a chunk of it, and then you have two CNVs that are actually the same but look different. And it's not the case that a given CNV had two separate individual origins that happened to start and end at exactly the same points. So it looks like, at least in most cases, a CNV allele represents one and only one evolutionary origin. Was there a second part to that question that I've forgotten? Or oh no, one thing I was going to add was that there is a slight extra challenge to looking at CNV haplotypes rather than the normal haplotypes, the haplotypes that Chris looked at for VGSC, which is that, so in order to do haplotype analysis, you need to do what's called phasing your data. So phasing, so each mosquito inherited at a given chromosome inherits one chromosome from its father and one from its mother. For example, chromosome 2, it will have one copy of chromosome 2 from its father and one from its mother. What we get when we sequence that mosquito is we just get the genotype, so AT, GT, TT, TC, etc. But we don't know which came from which chromosome. And the process of phasing is to identify, for example, okay, AGT, CATA, CGG came from one parent. We don't know that it's the father or the mother, but came from one parent. And then, these others, TT, that came from the other parent. That's the process of phasing. And that's not an easy thing to do. And CNVs cause problems with phasing because CNV makes the assumption that each individual only has 2 copies of any genetic region. By definition, where you have CNV, you suddenly have more than 2 copies of every genetic region. And as soon as you have more than 2 copies, it becomes very difficult to phase. And where you do phase, you might actually incorrectly phase. So that means that in these regions that have loads of CNVs, we can't get reliable haplotypes, and that means we can't get reliable phase data. We can't get reliable haplotype networks. So what we did for example, for the extended haplotype homozygosity, and the reason why there is this gap in the middle of the plot here, and the reason why these have been split into left and right instead of looking at the whole haplotype, is that we exclude the whole region that has CNVs in them. So for example, in CYP6AA1, we excluded the entire region covered by CNVs, and we looked, to the left and to the right of those regions, kicking out all of this. And so we're actually kicking out the data from the actual region that we're really interested in, and we're just sort of relying on flanking regions, and that makes it harder to build reliable haplotype networks. And so you have to sort of your associations with the CNVs won't always be perfect, and that imperfection does not necessarily imply that there isn't an association with the haplotypes. Hopefully that answers your question, Alhaji.
So we have about 5 minutes left. Sanjay has helpfully linked to a preprint in the chat.
Oh, I was going to link that later. Thank you, Sanjay. That's because it's hard for me to do with the talk. I thought you were about to say Sanjay has a question. I was going to say Sanjay, why now?
So yeah, I was wondering if you or Sanjay wanted to comment on this paper, of maybe also about what is so special about CYP6AA1, and what did you find in this preprint?
Yeah, I'm happy to do it unless Sanjay wants to do it, but I'm happy to go.
I'll let you go, Eric.
Okay, cool, thanks. Yeah, so this preprint is, I think the reason Sanjay posted it is that it's the one that talks about this DUP1 in Uganda. And so it's the one that has the evidence for the statements that I was making earlier about it's associated with insecticide resistance. However, it has other mutations that are linked to it and that we can't disentangle. So there's at least 2 other mutations that we identified: a SNP in CYP6P4 and a transposable element upstream of CYP6AA1. And because 2 of those mutations are always found together, one of them is sometimes found, and one pair of them is sometimes found on its own. But because we don't have some nice data where each mutation is isolated, we can't say, we found this effect, we found this associated with resistance. We can't ascribe that to any one of the SNP, the CNV, or the transposable element insertion. It's maybe just one of the 3, maybe all of them together, maybe just 2 of them. We just don't know. And so that's the main result of the paper, is that the CNV is important. Well, that this haplotype that carries the CNV, the SNP, and the transposable element insertion, this haplotype is important for resistance. It is spreading very quickly. So there's some really nice temporal data that shows an increase in frequency in the recent past. It's spreading geographically, because it started off, well, I don't know if we know exactly where it started off, but it certainly, you can see that it appears earlier in, for example, Uganda compared to DRC, but in both cases it increases in frequency. So it's spreading in time, it's spreading in space, and it's important for resistance, but primarily to deltamethrin and less so for permethrin, which of course in itself has implications for bednet distribution. So that's a quick summary of the paper, but it's a really, really nice paper by Haroon, who is now on a PhD program here, and who did this as part of his master's here as well. And I really encourage you to read it, because it's a very, very nice piece of work by Haroon.
Well, I think that's a pretty good ending point. Eric, thank you so much for presenting this work. And thank you everyone for coming and participating. Really great questions from everyone. So we'll see you at the next workshop. And as always, drop any questions into the Slack space or email specific people. Everybody's happy to answer questions. So
yeah, I would just say if you have any more questions on the paper, or as someone asked, if you want me to share the code or any of the background data, then please don't hesitate to send me an email. I'm not very good at checking Slack, so email, if you're directly addressing me, then email is better.
Okay, thanks.
Cool. Thank you, everyone.
Bye, everybody.
Bye.
Bye-bye. Thank you.
Thank you.
Thank you, Eric. Goodbye.
Thank you. Thank you. Goodbye.
Thank you.
All right. Bye-bye. Thank you.
Thank you, very well done.